Okay, so good afternoon, everyone. Okay, so last few lectures we finish up the nematode parasites, and we focused on the ruminant nematode parasites. So next two lectures I will give you a brief flavor of trematode parasites. So before we can start, I want you to let me know that which one is the trematode parasite in the next slide I'm going to show you. So just take it out your point solution and let's see which one is the trematode parasites here. Okay, so yes, so we so this A1 is a trematode, B we already know this is nematodes, and let's see in the cystode that will be the next one. Anyway, okay, so you don't need to be memorized that one, but now today we will be covered the phylum Platyhelminths. And the class is Trematoda. So the learning outcome of this next two lectures, you will get the understanding of the basic characteristics of the trematode biology, their life cycle, how we can diagnose these flukes. We will be discuss the treatment as well as preventive strategies of this group of parasite. We can also able to identify the drug of choices used for the treatment of these animals, and also the zoonotic importance of this group of parasite. So the general characteristics of trematodes: they are flat, bilateral, oral or ventricle suckers are there. So you'll see the oral sucker. So yeah, so you will see the oral suckers, and ventricle suckers are somewhere here. Incomplete digestive tract. They are hermaphrodite. Usually they do the self-fertilization, but sometime we have seen that they do the cross-fertilization also. So most of these species are dorsoventricularly flat. They are leaf shape, and they are covered with the teguments or cuticles. So you remember which stage of the nematode larvae has a cuticle? Sorry? Which stage? Anyone? Nematodes, I'm asking now, I'm talking about the previous lecture. Nematode, which larval stage has a cuticle? 3. Yes. So L3 stage. So same like here, this is a tegument is very similar to cuticle. These flatworms, they can protect themselves with this layer is outside the skin. So here is the general morphology of this parasite. You'll see the oral sucker here, the pharynx, oesophagus, intestine. Cuticle is the tegument, or cuticle is the layer here. Uterus, vas deferens, ovary, seminal vesicles, testes. So it means that one single worm, you will see the male and female sex organs, and they can produce eggs, and that's why we call them hermaphrodite. However, only one group of trematode parasite that have male and female sex organs. So this is really, really important for you to remember. Schistosoma is the exception here. So you can see the male and female sex organs, they are separated in and they can mate and produce the eggs. Is different than the other, all the trematodes, they are hermaphrodite except schistosoma. So adults of this group of parasite, they found normally in a number of different organs. Liver fluke, they are most important one. We will also talk about the GI tract fluke, lungs, pancreas, and veins. Schistosoma is in the veins. On the other hand, we will cover the cystode in the next lectures, where adults are always found in the intestine. And in this case, adults are found in a number of different organs, but the most important one is the liver flukes, and which we will cover, and the veins, the blood flukes. These are the most important one. Will be covered. Also, GI tract and lungs are also comparatively important. Okay, so what the egg of this group of parasite will be look like? They are always a heavy egg. It's not like a very small egg like nematodes. They are heavy egg. And you will see the operculum. This operculum always you will be see in the one side of the egg. So that is one of the identification point of this group of parasite to identify the eggs. For the life cycle, again, life cycle is really important for you to understand. If you really want to understand the clinical features, diagnosis, treatment plans, so the life cycle will be very important, and otherwise you will not understand the seasonal prevalence of this group of parasite, etc. Is indirect, always indirect life cycle. And snail are the first intermediate host. Some flukes have more than one intermediate host. Some has a one intermediate host, and snail is always the first intermediate host in this group of parasite. So what happened in the general life cycle of this group of parasite? Eggs pass out into the feces of the definitive host. They are unembryonated eggs. In the environment, they become embryonated. In the case of terrestrial life cycle, eggs ingested by the snail, and once the eggs ingest the snail, the miracidium will develop inside of the snail, and miracidium will be hatched there. But in the case of the aquatic type of life cycle, some fluke they are aquatic. We'll talk about that one in tomorrow's lecture. So the miracidium hatch from the egg, and then they penetrate into the snail. So there's two type of life cycle: terrestrial and aquatic. The difference is eggs is digested by the snail, and then miracidium hatch inside. So you can see this egg, and this egg will be hatched with the miracidium, and this is the miracidium, and this miracidium then go into the snail. So this is how the life cycle will be look like. In the next step, we can go a little bit more in detail what is going to happen in the snail once the miracidium go into the snail. So the asexual reproduction start within the snail. So one miracidium that goes into the snail become a large number of cercaries. How? First thing first. So this miracidium develop the sporocyst form in the snail. This is happening all in the snail. Then in the next step, these small reddies will be developed within this sporocyst. So this is all sporocyst. Once they burst, they further converted into this cercary form, and that is developed within the reddy. So cercary form is always have this tail, and that is happening in the snail. So from here, the cercary will be the snail will burst a snail, and the cercary will come out from the snail. Okay, so what happened next? Once the circari will be come out from the snail, so they can further convert it into metacircari form. So metacircari is, what is metacircari? This, yes.
Is mericircadian, is that just a term that's only used in trematodes, or is that
Is used in the trematode.
just trematodes only?
At the moment, yes, in the trematode. So this metacircari, once the tail will be removed, once this circari will be come out from the snail, the tail will be removed, and that is converted into metacircari form. The only exception is schistosoma. Schistosoma don't have a metacircari form. They will be directly penetrate into the skin of the host. This circari form will be directly penetrate. They will not convert it into the metacircari. So the metacircari normally, other than schistosoma, they can freely living into the posture, right? And then the definitive host will never get the infection from the snail. They will not eat the snail, and the infection will be go into the definitive host. Always metacircari, and that will be in the grass or in the environment, right? Anyone has any confusion here? Because life cycle is really, really important for you to understand before we can move forward. Is this clear? Okay.
So does it become the
Metacircari, yes.
so it does not become that until it's outside of the snail?
Metacircari.
Yeah.
That will be, the circari will be come out from the snail,
Okay.
having a tail. And then tail will be removed in the environment, and they convert it into metacircari.
Okay.
And this metacircari is eaten by the definitive host from the grass during the grazing,
Okay.
right? Okay, so this is what the general life cycle of this parasite will be look like. This is what we already discussed in the previous two slides. Adult, they lay egg. These egg have a operculum, and this egg will be further converted into miracidium, and this miracidium will be go into the snail, sporocyst, ready, and this circari. So these 4 stages are in the snail, and then this stage will be come out from the snail and converted into metacircari form. There is no tail, and then this was eaten by the definitive host and further converted into juvenile and adult stage within the host. And depends which parasite fluke is infected, and some goes into the liver, some goes into the lungs, but first they go into the intestine, and from there they penetrate into the different organs where they should supposed to be go. Yes.
So do they lose their tail inside the snail, and then the little mare or whatever comes out as the little, the metacircaria come out of the snail without the tail?
Circari will be come out from the snail. They have a tail.
It develops there, but then it comes out with the tail.
Yes.
Out,
And then tail will be removed, and they convert it into, yes.
okay.
Okay, so what about the snail? So these snails, they normally like this marshy pond areas. This is the best environment for these snails to be grow. So these snails die during the winter time once the weather is very very cold or either very hot. They like this. Their multiplication is more higher between 15 to 26 degrees Celsius. That's a very ideal temperature for these snails to be grow. However, they also go into the arrest state once and they burrow into somewhere in the muds. Once the weather is very much adverse or something very hot or very cold or very dry weather, so they go into the arrest state also in the mud. So this is the whether these snails will be the miracidium, the characteristics of the miracidium, how they can develop within the snail. So the miracidium develop immediately within, if the temperature is 10 degrees Celsius, and that is happening within the snail, right? And then miracidium develop with 2 to 6 weeks at once the temperature is between 15 to 20 degrees Celsius. So this is how the miracidium, they also need from, because remember we talked about this miracidium develop from hatch from the eggs. So they need the very favorable temperature to convert their stage from egg to miracidium. So this is the ideal type of environment they get to further develop their stage or convert their stage or hatch from the eggs. So in the characteristics of the metacercaries, so infective stage of nematode, which larvae is that? What stage of the larvae? L3, correct. Here, metacercary is the infective stage that can be infect the host, and then the host taken these metacercaries. That is always in the free living environment, and the infection only occur by the ingestion of the metacercaries, not snails, because they come out from the snail. Except, as I said before, schistosoma is the exception. The metacercary the cercary will not convert it into metacercaries in the case of schistosoma infection. These cercary will directly penetrate into the skin and go into the blood from the environment. So metacercaries are also resistant to dryness, but hot weather will kill them. So this is what the characteristics of the metacercaries will be look like. So now you know the characteristics of the cercaries and what is the characteristics of the metacercaries. Okay, so let's do some knowledge check questions here before we can move forward. Okay, so which of the following trematode has both female and male worms? Okay, so great, schistosoma. Okay, so which of the following stage is an infective stage in the trematode life cycle except schistosoma? So, metacercaries. So, now we will be covered some most important liver fluke of ruminants. Today we will be focused on Fistula hepatica or Fistula gigantica and Fistulidae magna. So, tomorrow we will be move forward with Dicrocilium dendriticum. Okay, so Fistula hepatica is a common liver fluke, large leaf shape, gray brown in color, and you will see the shoulder here is a bigger size fluke. You can identify from if you cut the liver, you will be see these flukes will be moving around in the liver. On the other hand, barrel shaped egg operculum is here, and this parasite reside into the bile duct, and the snail is responsible for the transmission of this disease. as well as the ruminant are the main host. Also, we have seen these parasite in horse, pigs, camels, as well as has a zoonotic importance in human. So, Fistula hepatica is very significant parasite as far as the production losses is concerned. Is a worldwide distribution. This parasite is highly pathogenic in the sheep as compared to cattle, but still causes disease, severe disease in cattle. Is very, very important and responsible for the production losses, and also one of the biggest cause for the liver contamination at the slaughterhouse. So liver will be rejected in the slaughterhouses if they will see the liver fluke infection in the liver. So that is one of the production losses. As you know, in some countries, they like African countries or Asian countries, they eat liver. So that is one of the production losses due to this particular reason. So the life cycle of Fistula hepatica. So I will let you know that this stage of this in the host. What is going on in the host once the metacercaries is taken by the host. So it will convert it into juvenile stage in the first instance, and that is in the liver parenchyma, less than 8 weeks. And then they convert it into immature stage in the bile duct between 8 to 12 or 14 weeks. And then finally they convert it into adult stage, that is between 12 to 14 weeks, and that parasite reside in the bile duct. Pre-patent period 60 to 90 days, and the full life cycle will be complete 5 to 6 months. So based on the life cycle we discussed, who gonna explain this life cycle to me? I put all the pictures here, make it easier for anyone to be explained.
So the egg gets shed from the main host.
Yes.
So then if that egg is eaten by a snail,
Mm-hmm.
which turns into the Vericidium,
Yes.
then the Vericidium becomes Revia.
Mm-hmm.
And then the Revia becomes the Ceria.
Yes.
The tail and penetrates outside of the snail to the environment, to
The metacercary. Yes, great. So this is how the life cycle, pretty straightforward, right? Okay, so now you can, I can check it out who understand the life cycle. So take it out your point solution, and we can go some questions and see if you understand the life cycle correctly here. So which fistula life stage infect snails? Okay, so yes, miracidium, the egg hatch into miracidium and they will be infect snail. Okay, so which fistula life stage is shed by the snail? Okay, so cercary. So remember, miracidium is converted into ready in the sporocyst, and the sporocyst it converted into ready, and ready is converted into cercary, and cercaries come out from the snail, and it is further converted into metacercaries outside the snail, and that is the infective stage. So, what is the name of the life cycle that insists on plants? So, yes, metacercaries, they are normally in the plants, and that is eaten by the definitive host. Okay, so distribution of the snail is the intermediate host. These are the states you will be seeing in the black color, the coast, Gulf Coast state, California, Pacific Northwest territory. So these are the number of different states where you will see the snail, Florida, Louisiana, or Texas. So this is the important in many areas of the world, including Canada or UK or other part of the world in Asia. So which species are present worldwide? So these snails are normally like the muddy area, semi-permanent pounds, and the most important species is Lamini of Formica, Fosseria in USA, and in the UK, Gulba truncatula is the most important species available of snail. So, clinical features. That is really important. In the case of cattle, if you will see the acute form of disease, young calves maybe show these acute clinical disease with heavy infection, and this parasite migrate, the larvae migrate into the liver and cause the traumatic hepatitis. So the larvae mean the juvenile stage of these fluke, and they penetrate into the liver capsule and responsible for the bleeding. So this is the acute form of disease seen in the cattle. So in the chronic form, there's a two type of chronic form, either subclinical disease. Normally you will not see any clinical prominent clinical sign, so there will be impact on the production losses, poor growth, and weight loss. On the other hand, in the case of the clinical disease, it is happen when the flukes are in the bile duct, and the adult worm start feeding the blood in there, and that will also further responsible for the anemia, hypoalbuminemia, or weight loss. So this is the two form of chronic diseases you will be see in cattle. However, in the case of sheep, it is much severe as compared to cattle. Is in the acute and chronic form, you will be see anemia or submandibular edema, anorexia, and weight loss. You will also see the ascites or death in the severe cases. So now we can see in the sheep, if you will see the anemia and submandibular edema. So why not you can immediately think about, we have seen that one in haemoncosis, right? So that's the differential point. So if you will be considered haemoncosis with anemia and submandibular edema, what will be your diagnostic test in that case to see if this is really haemoncosis? Because this is one of the other differential diagnoses here. So what will be your diagnosis? But what are you going to do? Before that, before fecal, yes, you will do it, but before that, pharmacia, and then confirm with fecal egg count. But here, you will not perform the fecal egg count. because fecal egg count will not going to work. This is a different technique, and tomorrow we'll perform the sedimentation technique you will be do for the identification of flukes, right? Okay, so as far as the pathology, this liver will be damaged due to the presence of these immature flukes, and they are one of the major cause of the liver contamination or liver rejection in the slaughterhouses. Also, death will also be occurred due to this liver rupture and the other disease, bacterial disease, Clostridium novii, that is a sequel of this rupture of the liver, and that will be this disease act on the liver once the liver is damaged due to this liver fluke infection. Okay, so this is the normal liver you can see here. But once this is infected by fistula, so you will be see some hemorrhages, and once you cut it down from here, you will be start looking near the bile duct. You will be start looking the flukes that will be moving around in the liver. So how you can see that? So look it into this thumb here. So once you cut it from here, you will be start see these flukes are moving around, and that will maybe you can easily have a look and see in your thumb that, oh, this is the liver fluke in the liver, right? Okay, so as I said, the methods are different. Diagnostic methods are different. You will be definitely start with the clinical sign, time of the year, and grazing history, time of the year. In the next slide, I will be show you the treatment plan, and that is really important for you to be remember time of the year. Which time of the year you will be go with the treatment plan also. So before that, we have seen the characteristics. They are upperculated, golden brown in color, large eggs, and you can perform the sedimentation technique tomorrow in your lab to identify the flukes. And this is a fluke finder. We will not perform this. We will use the sieve instead of the fluke finder in tomorrow's lab. But fluke finder is one of the other possibility. The flukes will be settled down once you cross from these fluke finders. So ELISA, one of the other method. So what you're going to see tomorrow, if you have eggs in the feces and you would perform the sedimentation, you will be see these type of eggs. They will be in the feces. These are the fluke eggs. Here you will see much bigger operculum is here. Okay, so now you have eggs. You can't be able to say this is the liver fluke eggs, or this is the intestinal eggs, or in the lung eggs, the eggs of the fluke in the lungs, because they are all come out along with the feces. Wherever they go, they finally come out along with the feces, right? So we developed this high throughput technology here, and if you give us a sample, we will be tell you which particular fluke species you have in your samples, instead of just saying this is egg. This is the egg, and potentially maybe a liver fluke egg, or maybe based on some morphology you can start thinking, oh, this is the intestinal egg or the ruminal eggs, liver fluke eggs. But this is the technology we have developed to be identify these high throughput method, and if there is a co-infection of number of different flukes, we can also identify that one too. Okay, so for the further, if you look it into the necropsy with the chronic disease, you will be see the lesions at the liver, something like that, and the flukes are also seen in the bile duct. Once you cut it down, you will see the flukes, and in the acute disease, you will be see enlarged liver with hemorrhages in the liver, and liver covered with the exudates or fibrinous exudates, and the small juvenile flukes you will be seen in the liver. So this is how you will be see in the necropsy of the animal. Okay, so treatment plan. That is really important to be remembered. Anthelmintic drugs in US: chlorsenal, albendazole. Chlorsenal, that drug is killed the adult fluke. This drug has a very poor efficacy against the immature and also the juvenile stage of the fluke. Albendazole, on the other hand, also kill the adult fluke and lower efficacy against immature and juvenile form. So it means that in the USA, if you want to treat the fischiosis infection, you have this choice. So you will kill the adult fluke. So in that case, timing is very crucial. So remember, we talked about in the life cycle, which particular time point you will be see the eggs, which particular time point you will see the miracidium, or which time point you will be see the adults in the liver, right? The best choice is the tricobendazole drug. That is a benzimidazole group, and this drug kill all the fluke stages. So what will be your preference then? Which drug you will be preferred in this case? Tricobendazole kill all the fluke. Chlorsenal kill only adult fluke. All fluke stages. Tricobendazole. And albendazole only kill adult fluke. So what will be your choice? Sorry? Yeah, go ahead. Yeah, you. Tricobendazole. So the problem here is you don't have that choice in USA. That's the problem. In USA, tricobendazole is not approved by FDA. And the answer is we don't know. So you have only these two choices here. Right? And this tricobendazole is approved in United Kingdom. So they're using there, but resistance is developing very quickly now. So in that case, time of the year is very important where you are working. So I'll give you example in Scotland. North temperate region, drug effective against immature fluke in autumn time. Drug effective against immature and adult fluke in the winter time. Drug effective against adult fluke in the springtime. So this is how they will be treated, because they know that which particular time point of the year they will see the immature and adult fluke, which time point of the year they will only see the adult flukes. So this is the example of one of the. So once you will be deal with this case, you should be know that what is the seasonal prevalence of this parasite in that part of the world. Otherwise, you have only one choice, that is tricobendazole, and tricobendazole is not a approved drug in USA for fischiosis. So here is the efficacy chart. Tricobendazole, all these stages, or other the clocental or albendazole, they are in the only effect on the adult stages. Okay, so control strategies. How you can control the liver fluke? First, snail. You have to think about how these drugs that can be, or this molluscides drugs can be killed, but they are prohibited. They are not going to use at least in UK. So the drainage, another way around, you have to think about how to control these drainage system, that water will not be stay in one particular area or one particular point where the snail will be start growing. In infection control, so how you can do that one, but before that you need to know how this infection built up in the host. So how many times this infection built up in the year. So this is what we have proposed, this multiplicity based genetic modeling. So we did this modeling and understand that how much time in the year the infection is built up within the host. So this is the basic understanding. Without knowing which particular time point you will be see these infection, you can't able to treat, because you know the treatment plan is different. You don't have tricobendazole. You need to know which particular time point the adults are, which particular time points is the mature flukes are. Grazing management also important. Not let the animal go into the wet area for a long time. So this is best area. The animal can be stay here. And also the treatment strategies. So you already know what is this treatment strategy. You don't have a choice of tricobendazole. So you have other choice. So then seasonal prevalence is important for you to be know that this particular season we have to treat these immature form or adult form. Okay, so let me ask you some knowledge check question here. Which of the following statement describe tricobendazole drug? Okay, so high efficacy against. So which of the following drug describe closanol drug? Okay, yes. Okay, so human fissulosis. As I said, they cause the zoonotic importance in human. More than 70 million people are infected with these flukes, and this flu, the fistula is the is listed as a tropical disease in WHO website. And normally the water curse, you will see this metacercaries in the water curse, and human can be eat this water curse, and the disease is transferred into human through these water curse. So this is how, and they complete the life cycle within the human. So once the human is infected, they have a prolonged fever, enlarged liver. They have is the epigastric pain, and this problem is very, very common in the US, Europe, Middle East, and Asia. Zoonotic importance is really, really concerning. Okay, so fistula gigantica will not go into the detail because the life cycle and biology and the clinical features, everything is very similar to fistula hepatica. The only difference is you will see the major problem in the Middle East, Asia, like India, Pakistan, China, and Japan. So this disease is common there. So let me finish up with the Fissulidae magna very quickly. This is a large liver fluke, at least 10 centimeter long, and they normally seen in the reindeer, in the Arctic, or in the north of Canada. And I can give you one example here. It's a very big liver. And we heard the news that from the native people, they eat these flukes from the liver, because they just put it into the fry pan, just fry it and eat it. So one of our colleagues, she went there and she have seen that one. So it's really, really interesting and surprising. So it's a very big fluke actually. Maybe tasty. Anyway, so life cycle similar to fistula hepatica, but the definitive host is deer. Sheep are the accidental host. Cattle and pigs are also a dead end host. So normally the distribution is seen in the Minnesota, Wisconsin, Michigan, and these are the number of New York, so number of different states where you will see this group of parasite. Highly pathogenic in deer, because what they normally do, they will be 2 or 3, for example, worms. They make a cyst formation and they will be seen within the cyst. This one is removed from the cyst, so even 2 or 3 worms can cause a very severe disease in deer. On the other hand, in sheep and goat, because they are accidental host, they are also pathogenic, and but in this case, flukes do not make a cyst formation. They just migrate in the liver, and again, few flukes may cause a severe disease and responsible for the production losses, as well as some cases if it's severe, animal become dead. In cattle and pig, because they are dead end host, mild pathogenic, normally you will not see the clinical signs. Some liver contamination, or the young flukes migrate into the liver, and the adult flukes, they encapsulate in a thick-walled cyst and no communication with the bile duct, so they will make these type of cysts in the liver. So diagnosis in sheep and deer diagnosis is based on the clinical sign and history, because they are very similar to other flukes. Eggs are very similar to other flukes, but definitive host is always the necropsy, because we've hardly seen the eggs of these flukes in the feces. And in cattle, because they are mild disease, you will not see the clinical sign. No egg will pass out into the feces, and they are accidental finding at the slaughter or necropsy of these animals. Treatment: albendazole for several days are the one of the choice here. Okay, so let me ask you one last question. Which of the following liver fluke primarily infect reindeer?
Are you saying that for this one, you can't see the eggs in the feces in the deer and sheep, or there's just no eggs at all?
No, you will not see the eggs in the feces without unknown reasons. Maybe they will not produce a large number of eggs, because the research, very little research has been done in this area. Okay, so yes, Fischelide